Hi, everybody. I'm Isaac Bradford. I am the owner of Filterponics, and I'm getting to do some cool videos with some cool people. And today we've got Ian Chesla with some research that he did in his master's program. So, why don't you go ahead and introduce yourself?
All right. Hey, I'm Ian Chesla. As Isaac said, I completed my master's degree at Purdue's Agricultural and Biological Engineering program just this past fall. I got to do a project on wetland mesocosms, taking a look at the denitrification and greenhouse gas emissions of different plant species in some mid-scale wetland ecosystems.
Awesome. Yeah, I got to see it in person, and it was really cool to see, especially, having done little bits and pieces. And so it was just really awesome to see this. And so I had to take the opportunity to ask you to do it again
Yeah.
for the internet.
Well, thank you. Yeah, I appreciate you saying that.
Yeah. So to kind of kick us off before we get into your presentation, I was wondering if you could tell us how do you choose this career path and why do you find this research or this area of engineering interesting?
Yeah. I think largely it was just following a thread of curiosity. I think the broad thing that got me interested was this idea of using ecosystems as treatment systems. I think nature is kind of this phenomenal engineer because evolution has kind of formed and sculpted it over, extreme like time scale. And we think about engineering of is like kind of this method of trying to like optimize certain systems. Nature has already optimized a lot of really amazing systems. So just kind of looking at how we can understand and maybe place some of those ecosystems as treatment systems. So using wetlands for water remediation and stuff like that. Just ecosystem engineering, I think, is really interesting. Yeah, really broadly, I think like biosystem or ecological
Hmm.
engineering. I know I really love ecology. I love being outside and hanging
Right,
out,
right.
and
Yeah.
I also really like problem solving. So I feel like it's
Hmm.
that intersection of like what I love and what I can do. So
That's
yeah,
that's
I
a
feel
good
like it's
point.
kind of ended up here.
Nice. Yeah. No, I definitely. Yeah, that's really cool. What you love and what you can do. That's a pretty good niche to find.
Yeah, yeah,
Yeah. Yeah. Cool. Well, I mean, however you want to present this, take it away. We're all ears.
awesome, cool. Yeah, so it has been a minute since I presented these slides, but we can definitely roll through them here and yeah, check them out. The title of my project was evaluating the biogeochemical influence of plant species on denitrification and greenhouse gas emissions in wetland mesocosms. So I think the project context is definitely like one of the more interesting parts about this. Just thinking about things like on a larger scale. Um, because I think research kind of funnels into like these very like fine details because realistically it's kind of like what you can do something about. It's something you can like understand and digest and use. When you get down to a unit that fine of like, okay, I know what this is and I know how to like put it somewhere and make it useful. You definitely like when you have one of those like units that you can actually handle. It's very fine. So um, thinking about the larger project context is really interesting because I, figured something out with this work, but it's like, where does it go is interesting. So, yeah, for the project context, expansion and intensification of agricultural production has resulted in a lot of excess nitrate runoff. So there's more people, they got to eat more food, so we grow more crops and also intensify the existing cropland. Um, and this results in more fertilizer use and in general, you get more nutrients that are running off from this crop production, nitrate, phosphorus. In the Midwest of the U.S., it's kind of that Corn Belt region. We can see the highest concentration increase of nitrate in rivers. So we're producing more food, even though there are a lot of there's a lot of advancement in agricultural practices. We still are seeing a lot of nitrate increase, and we're kind of experiencing some of these ramifications of using a lot of nutrients and having to grow a lot of food. And one of the problems that this creates, so these rivers in the Midwest are accumulating a lot of nutrients, and then those all funnel into the Gulf of Mexico eventually. And we see a lot of coastal hypoxia, the second largest zone in the world. There's consequences to ecosystem and human health. So essentially all those nutrients, they get dumped into this central location, and the algae there, they just take off because of those nutrients, and they're not like rate limited by those nutrients. So that's kind of like what would be their ceiling. But when there's a ton of nutrients in there, then they're going to explode up in population and in biomass. And then when they die off, all that algae is decomposing, and the decomposing bacteria that are digesting that algae consume a lot of the oxygen, so it's harmful for like the ecosystems. You kind of plummet in dissolved oxygen, and it's harmful for fish and other aquatic life. So why would you want to study wetlands? I've been talking about growing corn and fertilizer. It's like, where do wetlands come in? So these wetlands can be used to address the nutrient-based pollution in runoff. They're really cost-effective because an ecosystem is pretty self-managing. It's going to grow and change in the way that is like most effective for it to kind of grow and change. And an ecosystem is going to retain and remove a lot of those nutrients. So that nitrate, it can get captured by denitrifying bacteria. It can get assimilated to plant material. Phosphorus is something that's often sediment bound, so it's going to settle out in wetlands. Wetlands are going to help like slow that water flow, and things are going to be able to settle out a little bit better. So a little background on denitrification. Nitrate is what makes plants grow. It is also what can cause like some eutrophication in waters, because corn likes nitrate and so does algae. So if you're throwing nitrate on something, it's gonna get going. But this process of denitrification is where NO3 transforms into N2 gas. It's performed by facultative anaerobes in anoxic environments. So where there's no oxygen, the anaerobes are going to go ahead and take care of that nitrate. And wetlands that receive a lot of nitrate from ag runoff are going to be denitrifying hotspots, because you have a ton of nitrate and you have a ton of anoxic environment, because these wetlands remain continually saturated, so there's no oxygen really in the soils. With that being said, with those being pretty ideal environments, there is a pretty wide variation in how much a wetland is going to denitrify. So that's kind of interesting. Like, we have wetlands that can be used as these excellent denitrifying systems, but when we're looking at natural wetlands, restored wetlands, or just created wetlands, there is a pretty high variation in the denitrification. So if we wanted to kind of learn more about how wetlands can be used and how they can be used effectively to kind of mitigate this nutrient runoff, it's important to understand, okay, like, what's driving these differences? Like, why does this wetland really take care of business on nitrate, and this wetland is just slacking a little? So that's one thing. That's kind of one aspect of my research. And then the second big aspect is this greenhouse gas production in wetland soils. So anoxic soils, good because they promote denitrification. And then there's a drawback where anoxic soils also promote the production of greenhouse gases, with some notable ones being carbon dioxide, methane, and nitrous oxide. And this redox ladder on the right here, it essentially shows the most energetically favorable electron acceptors. So if there's an abundance of oxygen, the main metabolic process that's going to happen is going to be an aerobic process. Um, it's just it's essentially just taking like what's most preferred. Higher on the ladder is more preferred, and it's going to be the dominant metabolic process. It's going to be what you see happen with the microbes in the soil. So as oxygen gets used up, different things get used as electron acceptors, on a molecular level for these microbes. So talking a little bit more about these greenhouse gases of concern, carbon dioxide is a product of organic carbon respiration in wetlands. Methane accounts for wetlands account for about 1/3 of global methane emissions. So that is a pretty large amount for one kind of thing to produce 33% of global emissions. And nitrous oxide is the product of incomplete denitrification. So denitrification is great because it transforms nitrate into an inert gas in N2. But if this process of denitrification doesn't get complete on a molecular level, then N2O is produced, and it has a global warming potential of 296 times that of CO2. So essentially, it's a much better insulator of the atmosphere, which is a bad thing. And so we have these two processes going on. We have denitrification. We have greenhouse gas production going on in these wetland soils. What are some of the influential factors that drive these two processes? And it turns out many of the same things drive both of these things that we're interested in. So there's the carbon quantity and composition. So what is in the soil, how much of it is in the soil, and what type of carbon is in the soil. There's redox conditions like we talked about with that redox slider. What's available for the microbes to use as an electron acceptor? What are the dominant metabolic pathways going to be? And then there is nitrogen availability. So that one's pretty self-explanatory. Yeah, I was going to say something about that, but I'm like, well, that kind of sums it up. So taking a deeper look, how could we modify these influential factors? So we have wetlands. We kind of know what drives the things that we're interested in when it comes to wetlands. How do we change those drivers? How can we, kind of push this ecosystem in the direction of kind of doing something that we hope it can do? So if you think just kind of on a basic level, if you have a crop field and it's right by a river or it's right by any sort of water source, and you think, okay, there's a lot of ag runoff and it's going right into that water source. If we plop a wetland in the middle, that can help kind of mitigate some of that runoff. So then you're out there, you got your shovel, and you're like, all right, we're going to plant a wetland today. And deciding what you want to plant is a key factor that's going to come into any wetland design. So plant is a variable that you're going to have to decide on. And it is something that definitely has a strong control over both the process of denitrification and greenhouse gas emissions, because it drives those variables that it changes those variables that drive those processes. So soil carbon from root exudates and biomass deposition. So plants are going to push different carbons out of their roots, and also plants grow, they die, and they return to the soil. So they're going to deposit all the biomass carbon tissue back into the soil. Plants are also going to modify the redox conditions. So they're going to introduce oxygen into the soil. They're going to let gas from the soil into the atmosphere. And they do that through this parenchyma tissue. And it's basically like this straw-like tissue. So wetland plants need to get oxygen to their roots, but they grow in these submerged soils. So they do that by having like an increased parenchyma capacity. So they have this big straw that allows gas from the atmosphere, or a straw-like tissue that allows gas from the atmosphere that's touching the leaves to leak down into the roots, essentially. And this is going to change a lot by species. This plant tissue composition, grasses versus forbs, there's going to be some more structural carbons in some of the grasses to get them to stand tall. And then the parenchyma structure also changes a lot based on what the plant is. So you can see on the right here, there's the schizogenous parenchyma and the lysogenous parenchyma. That's a tough one. But the structure of that airy tissue is going to change a lot. So you're going to have an increased or decreased capacity based on how open or closed that tissue is. So we have these two processes, and we want denitrification to really take off and mitigate these nutrients. We want greenhouse gas to tone it down a little. So how do we balance these two things? And plant selection is a rarely accessible variable, and it is also something that is very effective. You can maximize denitrification. You can minimize greenhouse gas emissions through kind of selecting the right plant. And these plant species, they're going to modify these environmental variables, and they're going to interact in a complex environment. So kind of addressing how these plant species, these environmental variables, these interactions are going to modulate denitrification and greenhouse gas production is kind of the current knowledge gap that I'm trying to address with this research. So yeah, for the study that I did in these wetland mesocosms that I'll explain here in a second, I wanted to address the time scale and the effects of a large nitrate influx, kind of simulating agricultural runoff event to a big rainstorm, which fertilizer gets washed off into the wetland. How are these plants going to behave? The response of different plant functional groups, so grasses, forbs, is there going to be a difference based on like that plant group? And then also how the biogeochemical factors attributed to specific plants differ. So how does the carbon composition differ between plant A and plant B? And then look at specifically in my test wetlands, how said factors are going to impact the denitrification rates. Essentially doing the same thing for greenhouse gases, looking at the differences in production and emission among plant functional groups, the relationship between benthic production and system flux. So this is looking at how much greenhouse gas is being produced in the soil under different plant species, and then also looking at the system flux in terms of how much greenhouse gas is actually being emitted, per unit of wetland. And that one turned out to be really interesting because it's not necessarily like a one-to-one, where if you have a bunch of production in the soil, you have a bunch of emission from the wetland. There's some key factors that we'll get into that are pretty interesting that kind of help determine that. And then also looking at how biogeochemical factors attributed to specific plants differ, and how those factors are going to impact greenhouse gas, soil production, and system emission. So taking a look at the work that I did here, experimental design, I had 12 wetland mesocosms with 4 different planting treatments. So this was done in Indiana at Purdue, which is about an hour northwest of Indianapolis. And there was a depressional wetland on the study site that we were able to get our soil from. So we had developed wetland soil that we got to put into these big cattle tanks and run the experiment. So we used a couple different plants. We used reed canary grass, which is a common invasive grass in Indiana, and is linked to high denitrification. So it is an invasive grass. It's kind of everywhere. So it's obviously not something that you would go plant in your wetland, but since it is everywhere, it's worth understanding, giving it a look and checking it out. That's for sure. We also looked at rice cutgrass. It's a native alternative to reed canary grass. So this one has more design implications of like, all right, so you're not going to go plant a bunch of invasive grass. Instead, could we use this rice cut grass? Like, can we sub that out in some way and make it a little better? While also kind of maintaining some of like the high denitrification that people have observed in reed canary grass. And then we wanted to, those are two grasses, obviously, and we also wanted to look at the different plant functional groups. So we chose arrowhead, and it is another plant that is linked to high denitrification. It has a different parenchyma structure than the grasses. And since it is a forb, it's a flowering plant that has less cellulose than the grasses. So yeah, there's different carbon components in different plant groups based on like just plant structure and function. Grasses grow very tall and they have more structural carbon, so they can remain tall. And arrowhead is a much shorter, broader plant, so it doesn't need so much structure to, reach for the skies. And then we have a unplanted control for a baseline comparison. Experimental design stuff. We had a simulated agricultural runoff event. So we have nitrate. We increased the nitrate concentration in these mesocosms to 10 milligrams per liter. And we did this both in the growing and the senescent season. So we took some baseline measurements in the growing season. So in June, we did a nitrate pulse. We measured a lot of variables immediately post-pulse, and then 2 weeks after we performed the pulse to take a look at how are these systems immediately affected and how do they recover. We did this both in the growing and senescent season because it was important to look at plant activity versus plant senescence. And these systems aren't necessarily just going to be receiving a bunch of nitrate runoff, at an optimal time. So it's good to look at how they're going to perform, while the plants are growing, while they're not growing, at any time, really. So the environmental variables that we looked at, some of the measurements, we looked at soil, water, and the plants. We looked at water extractable organic carbon, and we also performed a KCl extraction to look at ammonia, nitrate, phosphate. We looked at total soil carbon through loss ionization and elemental analysis. We measured a lot of water parameters. Nitrate, UV absorbance at 254 nanometers, which measures this kind of more recalcitrant carbon. Ammonia, nitrate, phosphate, sulfate. We took a look at some carbon composition components, so some more detailed looks at carbon based on just like how much is there. We're looking at, what does that carbon look like? We did also answer the question of how much is there, so with total carbon quantity in the water. And we also did a plant tissue analysis, carbon, nitrogen, acid detergent fiber, lignin, neutral detergent fiber. What kind of carbons are in those plants? So with that, we can go ahead and take a look at some of the denitrification work. So how denitrification was measured was in the denitrification enzyme assay. Setting out for this research, we had multiple ideas for measuring denitrification. One of them was using some in situ cores that we could measure an N2 buildup using a membrane inlet mass spectrometer. And there just kind of ended up being too much troubleshooting with the instrument in order to get usable data from that. So it's very much an aspect of research is you try a lot of things and then like you hope at least one of them works. And fortunately the DA panned out really well. But yeah, it is it's kind of part of research is like, all right, we're going to measure one thing in like 3 ways. And one of them will probably work. So we had good success with this. We get the denitrifying enzyme activity of the soil under optimal conditions. And basically what the DEA tells us is the maximum denitrifying potential of a soil. So if this soil has everything it needs, how much is it going to denitrify? I like to think of it akin to knowing the top speed of a car. Your car isn't necessarily always going that speed, but you can compare upper limits. So I think it's a useful comparison, even if you're not getting, detailed like exact rates of like, okay, we're seeing exactly this much denitrification at this time. The comparison of like a Honda Civic to an F1 car is still really valuable. So you can understand like the max of different soils, the max denitrifying potential of different soils. So essentially this denitrification enzyme assay works by having a soil slurry in a sealed bottle, and you put a neutral headspace gas in there. You put nutrient additions into the soil so that no rates are nutrient limited. And then you inject acetylene. We talked a little bit about incomplete denitrification. This gas is going to stop the process stop the denitrification process at N2O. And this is because N2O is way easier to measure than N2 gas, because N2 gas is everywhere and it's hard to get accurate readings with most methods for quantifying that. So this DEA sampling, sediment was taken from 5 random spots in each mesocosm with a soil probe, and it was then homogenized and used for the DEA experiments. This data was then fed into a linear model. So we ran each mesocosm in triplicate. Here you can see we have mesocosm 1A, 1B, and 1C. It's 3 soils, all from mesocosm 1. And this N2O buildup over time is put into this linear model. We can generate a final rate of micrograms of nitrogen in the form of N2O per hour per gram of dry soil. So we can get this denitrification rate down to a unit of soil. Looking at the response of the nitrate pulse by plant. So we have this simulated agricultural runoff event referred to here as the nitrogen pulse. So when we dumped a bunch of nitrogen into the mesocosms, how did the plants respond? We quantified that with this delta DEA day variable. So we looked at the denitrification rates through the DEA the day after the nitrate pulse, and in our baseline measurements, we subtracted that out to look at the change there. And we also looked at the change between the pre-pulse denitrification measurements and the 2-week post-nitrate pulse measurements. So this addresses plants' immediate and long-term response to getting a bunch of nitrate dumped in there. And we can see that there are some interesting differences among the plants here. So in graphs A and B, you can see that the delta DEA day, how much denitrification changed from pre-nitrate addition to the day after nitrate addition. We can see in the growing season, arrowhead, the plant in the purple color there, was kind of above the rest of the plants. You can see it really kicked up denitrification in response to receiving a bunch of nitrate. And you can see that the 2 planted treatments, or the 2 grasses, remained above the unplanted treatment for that growing season in panel A. And then looking at panels B and D, during the senescent season, we can see that the denitrification goes down quite a bit in all the cases. So kind of having this warmer soil, this plant growth is something that is going to be a stronger influence on this denitrification activity. So looking at the response, again, further the response to the nitrate pulse by plant, the growing season response to the nitrate pulse, arrowhead had the strongest response. It was followed by reed canary grass, rice cut grass, and then the unplanted treatment. That change in DEA was significantly greater in the growing season compared to the senescent season, and the change between the pre-nitrate pulse rates and the 2-week post-nitrate pulse rates remained elevated in the senescent season, So we can infer that suggests a slower response or a slower nitrate utilization or a delayed response. So a bunch of nitrate comes into the system, and we see that nitrate is going to stimulate that denitrification action. And if we see that stimulatory effect remains a little bit longer in the senescent season. So there's no difference in the growing season pre-pulse and 2-week post-pulse rates for the DEA. And this suggests basically the nitrate gets used really fast when the plants are in their growing season. So within those 2 weeks, pretty much rates return to normal. Taking a look at some of these more detailed environmental variables. So what about these plants is driving this process? We fed all the variables that we measured into a principal component analysis. And basically, what we wanted to look at with this is how related are these variables? And a principal component analysis is a really good tool to use for that. You can look at a ton of variables and see how related they are by the angle between the arrows in the plot that the PCA produces. So the change in DEA the day after the pulse values were most strongly related to total water column dissolved nitrogen, stem density, and soil carbon. So we can see that this response to the nitrate impulse, the nitrate influx, is most closely tied to how much nitrate is in the system and how many stems are growing per area and how much soil carbon is in the system. And it has a negative relationship with SUVA 254 and UV absorbance at 254 nanometers. So the takeaway from that is less bioavailable carbon is going to have a negative impact on a system's response to denitrify a bunch of nitrate. And then that distance by plot is here. So you can see in that top right corner, SUVA 254, so measuring kind of that recalcitrant, less bioavailable carbon, and then in the bottom left corner, that delta DEA day value. And so we know, okay, so these are some variables that are associated with denitrification. Did they differ among the plants? And in a lot of cases, they did. So SUVA 254 is greater in the grasses compared to the other treatments, and you can see that especially in the senescent season. And then A254 or absorbance at 254 nanometers. Arrowhead is significantly greater than reed canary grass and rice cutgrass, which is pretty interesting because we saw that, hey, Arrowhead is this really good denitrifier. And so we can see in this A254 or the absorbance at 254 nanometers, Arrowhead has a significantly greater value than both reed canary grass and rice cutgrass, which is kind of interesting because earlier we saw, hey, Arrowhead is this really good denitrifier, and then we plot out all of our variables and we say, okay, absorbance at 254 nanometers when looking at carbon has this negative relationship with denitrification. So why does this plant that has a lot more of that carbon also show a really strong relationship with denitrification? And I think it's just that carbon isn't very usable. It's not necessarily that it really stops denitrification, but it's not going to help it. So it has a bunch of this unusable carbon that's not helping denitrification, but it also has a lot of usable carbon that is helping denitrification. So I think it's just like overall a much greater quantity. And then also we can see stem density. Rice cutgrass is way greater than all these other treatments. So there's going to be more stems per area growing. The rice cutgrass just goes really thick, and there's just more biomass, and that's something that would generally help with denitrification. It is more nuanced than just maximizing biomass, but we can see kind of some significant differences there. So kind of diving deeper into this carbon composition influence on DEA, we were talking about how certain carbons help, certain carbons don't help with denitrification. we can see that carbon influence on DEA is not purely based on quantity. So rice cutgrass is the most densely growing plant. It would contribute a lot of carbon to the soil just by this biomass deposition, but it's not the strongest denitrifier. And we can see here, there's a breakdown of some of the specific carbon components that we found. So not all carbon is equal in terms of how it's going to impact DEA. Certain carbons actually have opposite relationships. So we can see this component 1, this aged terrestrial humic-like organic matter, it's less usable for the denitrifying microbes. So we can see delta DEA day plotted on the y-axis. We see a negative relationship. So more of this humic-like terrestrial aged organic matter is going to result in less denitrification. We can see with component 5, it's tyrosine. This has a tryptophan-like fluorescence. We can see a generally positive relationship with that carbon component. So more of that carbon is going to help stimulate denitrification. Absorbent carbon, this A254 is aromatic carbon. We can see that has, again, a kind of a negative relationship. So this carbon that, like, the microbes are like, this stuff stinks. It's not gonna help denitrification. It's gonna go down. So overall, these less labile forms of carbon are going to have negative relationships with denitrification rates. And then that component 5, I think, is interesting because that tyrosine is not necessarily like thought of as like this like perfect carbon for like super bioavailable and like super labile. But there is some evidence in the literature showing microbes using tyrosine in environments where favored polysaccharide substrates are scarce. So I think that's interesting in the context of the mesocosms because these are kind of isolated systems, right? They're these cattle tanks. They're sitting out in the field. There's not like carbon inflow and outflow in the sense that there would be in a natural ecosystem. So I think, since that the inputs and outputs are kind of limited, those microbes are turning to that tyrosine. They're just making do with what's available. So we did see that positive relationship with tyrosine and denitrification in this case. Big takeaways: Arrowhead did a great job. But it was also the first to senesce, and it had the lowest response in the senescent season. So we see it just really perform well in the growing season, but it didn't perform well in the senescent season, and it dies off a lot sooner than the other plants in this study did. So that is important, framework just to understand. If you're trying to plant the perfect wetland, you might have a bunch of plants that really do great, but then they don't have the longest growing season, so they're not going to perform awesome for the longest. And the carbon takeaways, just kind of looking for plants with proportionally less humic or aromatic carbon and a little bit more labile carbon. That's going to help you reach your denitrification goal. So we learned something about plants, denitrification. That's kind of the first leg of this. And then the second leg is just looking at greenhouse gas production and emission rates, like we talked about earlier. So learning how much greenhouse gas gets produced in the soil, and then how much gas actually fluxes out of the system, like enters the atmosphere. So this is kind of a diagram that just shows all the things that we did. We measured the flux of greenhouse gases by using these gas buckets, basically. We just put a bucket over a floating bucket over the system, and then the water and the plants are going to release different gases, and they're going to build up in that bucket over the course of about an hour, and then we sample the gas over time. And then you can see on the right side there is this core. We had these clear acrylic cores that we pressed into the sediment, and we measured the gas buildup of what was emitted out of the soil over a period of time as well. A little bit of close-up on that core there. You're going to have benthic greenhouse gas production. So these benthic soils are just submerged soils. any soil that's underwater, kind of that first layer where the water meets the soil, benthic soil. Those gases are going to diffuse into the water overhead. And if we do it in a sealed system, we can measure that buildup and understand a rate of production from the soil. So when I say greenhouse gas flux, how is the gas actually getting out of, like, if we consider the wetland itself our system, how is the gas exiting said system? And it is essentially doing it through two main pathways. There's a plant facilitated flux. So we talked about parenchyma tissue a little bit earlier. Again, that straw tissue that lets gas come from the air or the atmosphere into the plant roots. It also allows gas to travel the other direction. So a bunch of greenhouse gas gets produced into the soil. It can be released through that plant into the atmosphere. And it's also going to flux out of the water. So a bunch of gas gets produced in the soil, it diffuses into the water, and the gas from the water diffuses into the air. We took a look at that system flux using the flux chamber or the floating bucket covering the plant in the water. Took samples every 15 minutes for an hour, used a linear model to calculate a gas flux rate, and then did some rate analysis looking at different predictor variables and how they could be used to look at that rate of gas flux. So we used two different buckets, one short one for the growing season because the plants were a lot smaller, they were kind of early on, and then we used some taller ones for the senescent season because the plants had finished growing and the grasses especially were really tall, so we needed some taller to measure the gas buildup. So what we did is we had a port on the top where we could attach a syringe and a excitainer, which is this little glass vial with a rubber septa on top, so you can pierce it with a syringe, and when you draw the syringe out, it seals back up. And you can vacuum down that container so that there is no background gas, there's nothing in there. So you know that when you poke a syringe in there and you put your air sample in there, you know that you have just a representative sample of what you drew up in the syringe and not like a bunch of extra background gas or anything like that. So gas from these floating buckets was drawn up into the syringe, and then it was expelled into this excitainer, and we could store those excitaners for analysis. So diving right into the data, we got the system greenhouse gas flux for N2O using a Tukey post-hoc comparison test. For the growing season, we can see the day after pulse is significantly greater than the day before the pulse. So this all kind of adds up. We know that adding a bunch of nitrate to the system is going to stimulate denitrification. It's also going to stimulate the production of N2O. So you have a ton of nitrate come in. Often when these systems get overwhelmed with a lot of nitrate, they're not going to, the process of denitrification isn't always going to be fully complete. It's just an organic system. There's a little bit of slop, if you will, that's just going to happen. So we do see nitrous oxide get stimulated from this nitrate pulse. For methane, there aren't statistically significant comparisons here. There is potential for noise in these data sets with ebullition, which is another process that I haven't talked about for greenhouse gas emissions. So if you have a big methane bubble that gets produced in the soil, it can bubble up through that water column and to the surface, and it can release a bunch of methane at once. So if you look at this top left panel for reed canary grass in the blue, you can see one data point is way higher than the others, and that is likely due to an ebullition bubble. So a bubble of methane came up through the surface into the bucket. We see this big methane spike. So it's not necessarily that data is bad. It's just that it is actually more representative of what is likely to happen, because ebullition is a realistic and valid process for this gas flux. For carbon dioxide, we did see some significant differences. Rice cut grass was greater than arrowhead in terms of carbon dioxide emissions. It was greater than reed canary grass, and it was greater than the unplanted systems. So overall, we see rice cut grass pumping the CO2 out. And then looking at the benthic production. So how much is getting produced in the soil? Not necessarily just how much are we seeing flowing out. What is actually happening in that soil? So we have those sealed sediment cores, and we measure the dissolved gas buildup in the water of those cores, sampling them every 4, 24, 48, and 72 hours after being sealed. This is a lot longer than the gas flux chambers because this diffusion is a slower process from soil into water. And in order to kind of get a grasp on a significant rate, you got to have a little bit longer time frame to see any appreciable differences. So again, we got a close-up of the actual cores on the left here, and then kind of a diagram functionally very similar to the gas buckets. The thing with these is we need them to remain 100% full of water at all times. If air were to get drawn into these cores, the dissolved gases that are sitting in the water would they would kind of flock to that air bubble. So it's like putting a big sponge in there, and it's going to soak up all of the dissolved gas. There's a lot more room in like a gas headspace for dissolved gas to be, so it's gonna kind of be all drawn up into that air bubble. And we did see some really interesting differences here. So with the comparisons, we see that arrowhead was significantly greater than the unplanted in the pre-pulse conditions, which are the conditions that we're really interested in. So just kind of at a baseline, do these plants have different methane production in the soils? And we can see that arrowhead is greater than all the other treatments, and through statistical significance is greater than the unplanted treatment, which is interesting because we didn't see arrowhead significantly greater in the system flux of methane. So we see arrowhead producing the most methane in the soil, but not fluxing the most methane out as a system. And then we also saw arrowhead greater than the unplanted systems for benthic production of CO2. And then rice cut grass was also greater than the unplanted for benthic production of CO2. So it's interesting that we have some discrepancy here, right? We have this arrowhead producing a ton of methane in the soil, but not actually releasing a ton of methane to the atmosphere. We have reed canary grass, maybe not necessarily significantly producing more CO2 or methane than the other plants in the benthic production, but we see reed canary grass and rice cut grass kind of releasing a lot of these gases through flux. So I think plotting out this way is really helpful to see. The benthic production is on the x-axis here, and then the surface flux is on the y-axis. So you can see in the purple with arrowhead, as the benthic production increases, we don't really see an increase in the surface flux. So it's a pretty flat line. But with both of the grasses, we see some pretty steep lines. We see that as benthic production increases in these plants, so does system flux. So benthic production does drive this system flux, but only in the grasses. And that is really interesting. Let's see, I think I get to it later, but so that suggests that the grasses have something about them that allows for that gas to be released into the atmosphere. Yeah, and that comes to the role of parenchyma here. So this total volume of parenchyma in a plant is going to depend on the structure of the parenchyma that's inherent to the plant species, and also the stem density. So how many stems you have growing per square centimeter. So maybe you have a plant that's pretty closed off. There's not a whole lot of parenchyma, not a whole lot of gas exchange going through one stem, but it grows really dense. So you kind of make up for it in numbers, and you have a lot of this gas exchange capacity. On the converse, you could have plant with a really open structure, maybe has less stems growing per area, but that really open structure is gonna, allow for more gas exchange. And this is what we found to be the factor determining the relationship between benthic production and system flux. So There's a visualization here of the rice cutgrass orencoma on the left. You can see it's got that huge open section in the middle, and then the arrowhead on the right has a lot more densely packed structure. So rice cutgrass, it's got a ton of stems per area. It's got a really open structure, so it's going to do it's like a super highway for transporting gases. In this arrowhead, it's got less opportunities for that gas to get from the soil to the atmosphere. There's less stems, and the stems that are there are a lot more densely packed, and there's not going to be a whole lot of gas exchange that gets through there. So you can see just kind of plotted out there, this rice cut grass, got a lot of stems per area. In the arrowhead has the lowest stems per area. You can see that rice cut grass has the second highest benthic methane production, while arrowhead actually has the highest benthic methane production. But we see the highest system flux for methane in rice cutgrass and the lowest system flux for methane in arrowhead. So it's this orencoma discrepancy that really determines if that benthic production is going to be relevant to the surface flux. So kind of just taking a look at everything here. This arrowhead is an awesome plant, right? Because it had the highest denitrification response, had low baseline and methane system flux. So you're like, yeah, that is awesome. And it is. It's a fantastic plant. But there's still some framework to it. I think in all ecosystems, there's no, superstar performer. There's no key that's going to solve everything. You think about application. Managed wetlands are ideally not monocultures, so you wouldn't want to go and just plant a bunch of arrowhead. You could run into problems. I think disease is a lot more problematic when you have a monoculture. One disease could potentially wipe out your whole system. I think ecosystems are also just beautiful things that exist outside of like a desired man-made function, and it's nice to see an ecosystem that is thriving with diversity and is doing its thing, as natural ecosystems do. And also on the side of thinking about some of these greenhouse gas consequences, there could be an interactive effect. So arrowhead has this high benthic methane production, so a ton of methane is getting produced in the soil for arrowhead. Think about what if you planted it next to rice cutgrass. So you ideally don't have this monoculture. You have these plants kind of intermingled. Potentially, could the high production in the soil of arrowhead be influenced by, or could that become relevant if it's planted to a plant with a lot of capacity to facilitate gas exchange like rice cutgrass? Additionally, like we talked about, arrowhead was one of the first plants to senesce. So you would want to kind of consider incorporating plants that grow maybe earlier than arrowhead or later than arrowhead. You can extend your wetlands growing season and kind of increase your overall denitrification. So yeah, these plant rates in greenhouse gas emissions and in methane, in denitrification and greenhouse gas, the rates are really important to understand on an individual plant level and an ecosystem level. It's important to understand how all of your parts fit together. And in order to understand how they fit together, you got to understand how they function on their own. I always think of it as you can have like a really stellar soloist, and finding that soloist is amazing and it is really important. But it's equally important to understand how that soloist might fit into a larger orchestra. I think that's really how ecosystems are. They exist in orchestra, and they become fantastic through this kind of cohesive melding of a bunch of different moving parts. So yeah, there's a lot of folks along the way who have helped this research, and many, many more who aren't listed here that I'm very thankful for. And yeah, I believe that is my presentation.
Awesome. That's great. Great job, man. Yeah. It's
Thank you. Yeah, it wasn't too long, but
oh no, all good, all good. If people are really worried about it, they can scan through and find the pieces they really want, but they should take a chance to go through it because I mean, I know it was an early morning when you gave the presentation the first time, so it's really cool to see it again because I definitely took away from it more this time than I did the first time I listened to you talk about it.
nice.
And I've got I mean, because, you know, we have our audience here. Actually, we don't, so that's the issue. So I figured I'd ask some questions, perhaps, at least stuff that I'm interested about. And, you know, I can leave, you can, if you want to put an email in or something, I can also write that down in the description if you want people to come talk to you about it. But I was kind of wondering, so you talked about a lot, right? You talked about, you know, you've got stem density, denitrification, carbon, all of these things that play a role in everything. And as we were talking about before we started recording, kind of like how academia does play a role in advancing the world, right? And so for your role, I'm kind of thinking like, you know, we've got these wetlands a lot of times, you know, especially in the Midwest, you know, where we've lived basically our entire lives. A lot of places had wetlands in the past, right? They had those symphonies that were helpful and everything, and now we've turned them into agricultural and farms, and we do have this sediment, right, that is eroding off of fields. So I guess before I get into my next phase of the question, like how important is this relationship between sediment and phosphorus, and how does that play a role in your research?
Yeah, so I guess as I've understood it, phosphorus is often a sediment-bound nutrient. So as sediment kind of gets washed off from these fields, it's taking a lot of phosphorus with it. So there's ramifications for losing phosphorus from your cropping system, and then also introducing phosphorus into a system that it's not native to necessarily in the quantities of like sediment-rich runoff.
Gotcha.
For my research, I didn't look too much at phosphorus. I focused more on nitrate just to kind of get it down to like a more digestible unit.
Right.
But it definitely would be a great thing to look at for some future research.
But a plus just for the wetland in general, right? Just because like you talked about briefly, which I just figured you could mention it again, is that wetland does catch that sediment. So on top of all the things you've already mentioned, that's an addition as well, right?
Yeah. Anything that's bound to the sediment can kind of help it can be settled out by just having a wetland there to slow the water flow rate.
Right. And I think there's I think there's some research with the pathogens like E. coli or something being bound to like very fine, like settling to the bottom, like not necessarily as sediment or sediment bound, but interesting stuff.
Yeah, that's really neat.
Yeah, but I was gonna ask with that, like after the sediment is like, so that's important for farmers, but also so is nitrogen. So you've talked about creating this orchestra of an ecosystem, and how does that fit in for a wetland? It's like if you're gonna talk to a farmer, like what would you suggest for like sort of a planting based off of your research, or sort of like a location, or sort of like is a wetland like a fix-all solution, or is there some things that we need to take into consideration when doing like edge of field management practices?
Yeah, I think it's dangerous to call it a fix-all solution because I think there is a right context for it. You wouldn't want to try to force a wetland in where it's just not naturally going to fit and thrive.
Sure.
I think having a wetland function as a healthy ecosystem is the key to getting it to kind of produce the results that you're looking for. So if you have, a field that's surrounded by like these really deep, narrow, like fast flowing drainage ditches, then maybe a wetland isn't going to be perfect to put down there because you're going to be forcing a bunch of water through it. You're going to be washing out a lot of the plants that might not be able to grow. I think I've heard about the idea of like fractal patterns in like permaculture when people make really fancy gardens in their backyard. And it's this idea that like, you can make one system. How does that system fit in with other systems? And then that group of systems makes like a larger system. And how does that group of systems fit in with like another group of systems? visualized in fractal patterns. If you think about like a rhombus, you can fit 4 rhombi together to make a larger
Right.
rhombi,
Okay. Yeah. Yeah.
and
Yeah.
then you can fit those rhombi together.
Gotcha.
So I
Gotcha.
think that wetlands, they kind of work in this fractal pattern thinking of okay, we have this crop field. In a wetland is going to be really applicable here because we kind of have like this slow runoff. We have like a natural depression area. If you put a wetland right there, it's going to be awesome.
Right. Yeah, because I mean, now that you kind of say that, like you're talking about the fractal patterns, right? Like, so if you had a field and you had a wetland, but the guy next to you also has a field and a wetland, like you start managing it as a network, right? Which is kind of like what it used to be, right, with oxbows sort of thing, where the land kind of managed itself. Well, managed it through not managing it, more or less.
Yeah.
Yeah. A little bit of chaos for order, sort of thing, I guess.
Yes. Yeah.
Right. Yeah. Cool. Yeah, I guess because the whole greenhouse gas is like, maybe that's not like necessarily something that you can make money off of, right, as a farmer, but people are still interested in it becoming a bigger and bigger conversation every day, all the time. Do you think there's a way with some of those EPA funds to have wetlands or some of those additional funds for like wildlife, right? Because they do these wildlife restoration. You can take a segment of your field and put that there. Do you think that there's enough, pros and money that you think it's a good idea that we could start instituting these things on a wider scale?
Yeah,
I know it's a little out of the scope of what you talked about, but I just think like big picture wise.
Yeah, I can touch on it a little bit. I think I don't know if this addresses your question exactly.
You just take it away wherever you want to take it.
Yeah. I think one of like the big economic benefits of instituting a wetland is it's mostly self-managing. So you plant this wetland, that's obviously like an upfront cost,
right?
To the planting, getting the plants. If you have to do any like earth moving activities, you have like an upfront cost there. And there is this management period, especially after the first couple of years while the wetland is establishing itself. If you have a big plant die-off, that's something you got to address. If you have different invasive species coming in and really taking over the wetland, that's something you also have to address. Gotcha. But once that wetland is established after the first few years, the amount of time and money that you have to put into it is
minimal, more or less.
Pretty much nothing. Yeah,
Gotcha.
It's doing its job just by existing.
Okay.
So I think there is an economic feasibility there and an appeal there in that kind of self-management. I think as far as like getting other funds into like creating wetlands, I think there's definitely opportunity for that. Wetlands are just like, aside from like functional ecosystems, they're just like very like biodiverse and beautiful. And there's like, different, birds and fauna that love wetlands. So there might be an appeal there to kind of get some preserve sort of money into wetlands.
Right. Cool. Awesome. And I guess this is kind of my last question. It's more of an idea, and I guess if you could explain it a little bit more. So it sounded like based off of the 2 chapters that you had, kind of arrowhead and rice cut grass kind of stood out the most, right? So do you think if you were doing like a medium-sized wetland, like balancing those two and maybe some, flowering plants for biodiversity, or I know that some people do brush maybe on the sides, kind of like as depending on, how bad wind is, or maybe if you're trying to have a lot of extra brush to have, against water runoff, like is that like a good combination, is to get that the stem density and the balance of the methane from the arrowhead and everything else?
Yeah. So I feel like it's kind of a tough question to answer because it is also like a situation specific thing that you would want to address. If you think about, I don't know, there's I've heard of the idea of using like a sediment forebay in a lot of different wetlands.
Okay. Yeah, that's kind of right. Yeah, something to block it off first, and then okay.
Yeah. So you definitely, I think there's definitely some room for like a really neat system where you have like some more robust plants in a sediment forebay, slow things down, and then you have maybe like some arrowhead in the center when your water's not flowing so hard, and that can be like your denitrification hotspot.
Gotcha.
And hopefully a lot of your nitrate will settle towards that area.
Right.
Yeah, as far as what plant species you would kind of want to like plant together, we talked about like the rice cutgrass and the arrowhead. I think there's more work to be done in terms of like understanding some of those interactions.
Right. Well, that leads me to my next question. So for our like last couple of questions that are just kind of in general, for the area that you've been working in, and for like up and coming research within the topic, what do you want people like? What do you want to see people research, or what do you want to see discussed more frequently?
Yeah. So I think for one, it'd be interesting to look at a couple more like plant functional groups, and just kind of understanding the denitrification and greenhouse gas production of different plant functional groups, and also kind of advancing this idea of looking at different plants with different levels of carbon and seeing if how far we can kind of push that boundary. So we saw this tie of labile carbon to like higher denitrification. How much can we kind of advance that? Like, what's the plant that we can use that has the most, bioavailable carbon?
Sure.
And is there a ceiling for that? Do we see like a diminishing return somewhere, or is it pretty much just a continuous relationship? I think that would be really interesting to see if we're interested in maximizing denitrification. We kind of found this variable that it seems like can be used. But how far can it go? I think is an interesting question.
So kind of for somebody to pick up the torch and keep running with it.
Yeah.
Nice, very nice. Well, hopefully someone sees this and does that too, or maybe there's already someone out there doing that and just hasn't published it yet.
Yeah.
It'd be really cool.
I'm sure there's. Yeah. It's somewhere out there.
Somewhere. Hopefully, maybe it's just not in the language that we speak too. That could be part of it.
Could be. Yeah.
So I guess now that you've got a job, right? How has your research experience like helped you with your career or picking a job? Because sometimes finding out, like going from undergrad, maybe you're like, I'm not ready for a career, but I want to do master's degree. I mean, everybody's story is different, but finding out what you don't want to do is sometimes the best way to find out what you want to do. So I guess, how is that experience, and how is that transferred over to your job now?
Yeah, I think like maybe in like a less conventional way of like, I took these details and this knowledge and I can apply it directly to my job.
Right.
I think more so just of like going through the master's project. It was like, okay, like I have this study, this project, and like I have to complete it in order to finish this degree. So I feel like I found myself in a lot of like scenarios where it's like, okay, this project isn't quite going as I planned. I have to fix it. Like I have to figure out how I can live with this data gap. I have to figure out how I can fill in this data gap. I have to figure out how to synthesize something I don't understand. I think, I think just kind of being met with scenarios that are unfamiliar or challenging, and then kind of the only way out and being through them is something that I've been able to take into my job and just say like, okay, and not like freak out like, okay, I don't get this. Like, I'm just like, okay, I don't get this. I'm going to get it.
Yeah.
Like, it's like I've been here before in a situation where I've been confused or overwhelmed, and it's like, okay, I'm going to make it through this, and I'm going to come out on the other side. Like,
I ask you a question, you don't know it now, you'll figure it out, sort of.
Yeah.
Nice. That's pretty good. That's a pretty good, learning step for sure. Hopefully I get to that point too, that I can, a lot of resiliency, I'm sure, in your years of grad school.
It's just a process.
Yeah.
It just, I don't know. It builds over time too. I'm not like, I still, get overwhelmed and have to figure things out, but,
being in a grad school office is definitely a newer experience.
Yeah.
Yeah. I guess this is kind of a looking back sort of thing. What is one short piece of advice that you would give yourself, like maybe, kind of from like undergrad to now, looking, because like you're at like that new stepping stone of your career. What's a piece of advice you think would be helpful?
Yeah. That's a good question. I think that it is good to uh, be resilient in your work, um, and just kind of like spring back from your setbacks and not kind of be crumpled by them. And understand that there are going to be times where you kind of fumble the bag, and that's okay because you can always kind of pick back up. And also just kind of being open to learning about a really wide variety of things. I think having your own enthusiasm for learning is really important, and sharing in others' enthusiasm is a really beautiful thing too when you can. So yeah, yeah, that's what I have off the dome, I guess.
That's pretty good. That's pretty good. I like that. That's nice. That's nice. And then my final question, maybe the hardest question, depending on how you see it. Uh, Kind of for fun. What is a plant or animal? If you want to pick a fish, that'd be cool too. Which is still an animal, but do you think is cool, unique, interesting, means something to you? Whatever comes to mind first.
Yeah. I really like I did a lot of bird watching. I
Okay.
haven't I've been out of the game for a couple of years, but I really enjoy sandhill cranes.
Okay.
Um, just kind of these big, elegant, graceful birds,
Mm,
and they have a really beautiful call. And it's always really nice when you're working outside and like you're just like in the morning, you're working outside, and like things are kind of quiet, and you hear like some sandhill cranes going overhead. It's like, all
uh,
right, it's
nice.
beautiful.
Nice. Good pick. Good pick.
Thank you. Yeah.
Yeah. Cool. Well, I guess, is there anything you want to say or put out there, or anything you think people should look up and, you know, research for themselves? Anything you find interesting you want to share?
I think just following your own threads of curiosity. That's probably the best thing you can do.
Cool. Cool. Well, a big thank you to you, sir. You did a great job. It was
Thank
awesome
you
to
very
see
much
you
for
again.
having me. Yeah,
Yeah. Cool.
I appreciate it.
Yeah. Well, alrighty. We'll catch you guys next time.